Back to our first session today in the conference. This session will be headed by my colleagues, Professor Safa Shafi, Professor of Pediatrics and Neonatology at Shams University, Professor Ghada Gad, Professor Shams University, and Professor Hatem Saafan, Professor of Pediatric Surgery, Pediatric Surgery Department. The session is entitled Learn from Ligands in Neonatal Gastroenterology. And as the agenda shows you, it will run in 2 successive lectures, and each lecture will be followed by a brief discussion. Then we will have a scientific symposium. Let's quickly introduce and welcome our distinguished guest speakers from the US. We are really greatly honored by their participation in our conference today. We have Professor Josef Neu, Professor of Pediatrics, Neonatal Prenatal Medicine, University of Florida, and Professor Jack Sek, Professor of Surgery at Harvard Medical School. And now I will leave the talk to my dear colleague, Professor Ghada Gad. She is going to moderate the session. تفضلي دكتورة غادة.
Thank you, Professor Nihal. يعني أستاذ أحمد, I just want to عايزة أتأكد بس إن الناس دخلت already ولا لسه؟
تمام يا دكتور, اتفضلي. الناس
يعني
موجودة.
already خلاص. طيب تمام. Okay. Good morning, dear professors and colleagues. We are honored and delighted by the presence of Professor Josef Neu, University of Florida, USA, the legend of neonatal gastroenterology. Professor Josef is internationally recognized for his research in developmental gastroenterology and nutrition, and most recently focused his research efforts on the intestinal microbiome and mucosal immunology. Professor Josef received during his career several honors and awards. He chairs and is involved on national and international committees. He's active on several society membership, editorial boards, journal reviewer, service to the community, and service to schools. Professor Josef has received many appointments. Last one was Associate Professor and Associate Division Chief for Neonatology, College of Medicine, University of Florida. He's Director of the Neonatology Fellowship and Director of Neonatal ICU. Professor Josef, I know how busy is your schedule, and I appreciate your contribution, especially today where you have many obligations. I thank you very much for accepting being with us this year. And this is the 3rd year you cooperate with us in our webinar or in our conference of Ain Shams University Neonatology unit. Actually, we are honored and delighted by your presence. And now it's time to listen to the legend of gastroenterology in neonates, Professor Josef Neu. Professor Josef, you can share the screen now.
Yes. Thank you very much for that kind introduction. I'm really honored to be involved with this conference, and it's always a pleasure to be with my friends in Cairo and beyond. Over the next 45 minutes, I'll be talking about the developing microbiome in the perinatal and neonatal periods. And here's my disclosure statements. I have no conflicts of interest that relate to this lecture. So over the next 45 minutes, I'll try to provide a little background and review of the developing microbiome, both prenatally and postnatally. I'll talk a little bit about the early life intestinal microbial interactions, some environmental factors such as nutrition and antibiotics that can affect microbial intestinal interactions. And then I'll talk about multi-omics, and as we go through this, we'll also have some clinical correlations. So let's begin over here. How's this? Can you see the screen okay?
Yes, sir, we can see.
Okay, so this was the agenda that I just mentioned. And so here we have a picture of a person by this body of water. So if any of us were going to go to a river or to a lake or to the ocean and take a drop of water, and if we take that drop of water and put it under the microscope, we would see lots of microorganisms. However, if we take that same drop of water and put it onto an agar plate in a petri dish, we probably would not see very many of these microorganisms. This is something that is called the Great Plate anomaly, because we are not able to culture all the microbes present in a certain niche, such as a lake, the ocean, a river, or even from our own bodies. Now, the history of the microbiome actually dates to the Human Genome Project, where in the late 1980s, early 1990s, the large project called the Human Genome Project started. And with the Human Genome Project, the goal was to sequence the adenine, thymine, guanine, and cytosine of the human DNA. And over a period of about 10 years and many billions of dollars later, this was successfully accomplished. The human genome was successfully sequenced. And here are just some of the covers of medical journals and also popular press journals that showed that we had finally cracked the human genome. Now, along with the cracking the code of the human genome, a lot of new technologies were developed. And these technologies involved something that went well beyond looking at trying to culture bacteria in agar plates, as we see over here. If we try to culture bacteria from our gastrointestinal tract and use lots of different kinds of agar types of technologies, we might be able to culture around 400 species. But with these new DNA sequencing technologies, we are able to find over 2,000 species in our gastrointestinal tract. And so this tells us that we are opening up a whole new world using some of these sequence-based technologies to be able to evaluate microbes. So here are a couple of these sequence-based technologies. And on the left, we see something called 16S RNA sequencing. And what is done with 16S RNA sequencing is that a small part of the microbial genome called the 16S segment is chopped up into small pieces and then realigned. And then these realignments are placed into a computer that tells us what kind of sequence is present. And it also tells us the diversity of microbes. It tells us the relative abundance of species within a sample. This is now becoming relatively inexpensive. And so 16S sequencing tells us who is there, what kind of taxonomy is present in a sample. A much more expensive technique, and one that involves whole genome sequencing of the microbiome, is one that tells us a lot more information. It tells us not just who is there, what kind of taxonomy is present, but it also provides us with some kind of knowledge about what the capability of those microbes is. So it tells us not just the names of the bacteria or their relative abundance, but it tells us what they have the capability of doing. So we're beginning to recognize more and more that the human is more than just an individual who is derived from the sperm and the egg, but we also have another component, which is the microbiome. And when we compare the genes that we have that are derived from the sperm and the egg, there are 23,000 genes. But if we also add the biome, that adds 3 million microbial genes. And so this is why we are called a superorganism that not just it consists of the human genes, but also the microbial genes that are involved. Now, I want to introduce you to a term called the holobiont. What is a holobiont? Well, it can be defined as a super meta-organism that adjusts and transforms itself according to environmental change, causing evolution of the entire entity. Now, here on the left side of this slide is something you might see if you go scuba diving or snorkeling in the Red Sea, this beautiful coral reef. Okay, a coral reef is made up of invertebrate animals, microbes, algae, fungi. It's an ecosystem. And if that ecosystem is perturbed in a negative way, this happens. This is called coral bleaching. And we've been seeing this happening now in many of the coral reefs around the world. Why does this happen? Well, with the warming of the oceans, something called global warming, the pH of the ocean changes slightly. And with that slight pH, that changes the microbes in this coral reef, so that from this beautiful, colorful coral reef, we have this bleaching. So this is telling us that small environmental changes, not just in coral reefs, but also in our bodies, may have an effect overall that causes evolution of the entire entity. Now, in our gastrointestinal tract, we have microbes, and the microbes and the number of microbes and the types of microbes differ depending on the region of the gastrointestinal tract. In the upper gastrointestinal tract, we have very few bacteria. But as we go down in the gastrointestinal tract to the more distal region, to the ileum and to the colon, we start getting an increase in the number of bacteria, so that in the ileum we might see about 10 to the 8th, in the colon 10 to the 12th bacteria per gram of luminal material. Now, the function of the bacteria and how it interacts with the intestine is different depending on the region of the intestine. So in the ileum, we see a function that is more of an immunologic function, and in the colon, we see something that is more of what we call a metabolomic or bioreactor function. So what do we mean by this? Well, the metabolomic or bioreactor role is one that can be represented first by this animal that we see here. This is a termite. Some of you may be familiar with termites. I know that where I live in the southern part of the United States, if you have a wooden home, it is very dangerous for this home to be infested with these termites, because termites are able to eat wood. They're able to eat and digest wood. Wood is actually a complex carbohydrate. Why are these animals able to do this? Well, they have enzymes in their gastrointestinal tract called lignases, which help, but they also have a certain microbial system in their gastrointestinal tract that helps them digest this very complex carbohydrate. Now, in people, here you see a very fat individual, here you see a very thin individual. If you look at the microbes in this person's gastrointestinal tract, they will likely be very different than the kinds of microbes in this person's gastrointestinal tract. And it's been found that if you take some of the feces from this individual, separate out the microbes, and put them into a germ-free mouse, early on in life, that germ-free mouse will look like this. If you take this individual's microbes and put them into a germ-free mouse, the germ-free mouse will grow up to look like this. So the types of microbes in our gastrointestinal tract can actually have a major effect on development of our metabolic capability and also our obesity or thinness. So here is an example, a picture of how this can happen. Here's our gastrointestinal tract, and nutrition, drugs, and other environmental factors can play a role directly on our gastrointestinal tract and affect how the human genome is interpreted. But intermediate in our gastrointestinal tract are the microbes. And we feed these microbes with the nutrition, with drugs, and we affect the microbes through antibiotics and various other factors. And these can have effects on the transcription of DNA using epigenetic effects, and it can also have effects on the RNA, silencing or enhancement of protein production. So this is part of the metabolomic role of the microbes. In addition to the metabolomic role, there's an immunologic role. And it was found in the early 1960s that if you take conventional mice raised in a conventional cages that are exposed to microbes, and if you also raise germ-free mice, and if you take their axillary lymph nodes and look at them under the microscope, the germ-free mice have these germinal centers that are not very well developed compared to the animals that are raised in a conventional environment. So this is telling us that exposure to microbes helps development of the immune system. Now, I want to take just a few minutes to go through some of the history of development of the mucosal immune system in our bodies. And here we have the ileum and the colon, and I want to focus here on the ileal intestinal barrier. In the ileal intestinal barrier, we have numerous cells that line our gastrointestinal tract. Some of these are just the regular enterocytes, and these enterocytes are involved in absorption of nutrients. We have several other cell types called goblet cells. These secrete mucus, and that mucus helps to immobilize bacteria. We also have these Paneth cells in the bottom of the crypt. These secrete defensive materials, defensive proteins that help protect this highly mitotic region of the gastrointestinal tract. This is where cells are born, and these cells then migrate to the villus tip, and they're extruded from the villus tip. But if you don't have these defensive molecules, these cells in this highly mitotic region will die, and the intestine will no longer be able to function. We also have cells that produce immunoglobulins, and these immunoglobulins are very important in homeostasis of the gastrointestinal tract and the mucosal immune system. Now, back in 1985, this person, Professor Christian Volhard-Nusslein, was doing some studies on this animal here. This is the Drosophila, and they were doing developmental biology studies. What causes mutants of this Drosophila to develop? So sometimes you saw a mutant where the legs were actually coming out where the antennae would be. And so they were looking at the protein differences in the mutants versus the wild type animals. And one day, one of Professor Nusslein-Volhard's graduate students looked at a protein gel from a mutant Drosophila versus a wild type Drosophila. And brought this to Professor Nislein-Bohlart, and Professor Nislein-Bohlart said, "Das ist ja toll." In German, this means, "This is really awesome. This is really cool." That's what the word toll means in German. A few years later, at Yale University, Professor Charles Janeway was doing some studies on macrophages, trying to determine what kind of proteins in macrophages interacted with microbes. And he found a certain kind of protein that looked very similar to the protein that Professor Nislein-Bohlart found in the Drosophila. Well, he found this protein in macrophages, and then these proteins were subsequently found on the surface of the intestinal epithelial cells. And now we know that there are receptors that are these toll-like receptors that are found on the surface of various kinds of cells. There are about 12 of these toll-like receptors, and these receptors interact with microbes and components of microbes. So, for example, here we have lipopolysaccharide, which is found in very high quantities in gram-negative microorganisms interacting with this toll-like receptor 4. Here we have toll-like receptor 2 that interacts with lipoteichoic acid from gram-positive microorganisms. And these all cause a transduction of signals that go into the nucleus. And in the nucleus, you have transcription of messenger RNA that causes the formation of an inflammatory response, which is very important for fighting off infections. Now, here's some more historical information. These three individuals all won a portion of the Nobel Prize a little bit over a decade ago. And this person here, Ralph Steinman, won 50% of the Nobel Prize. These other two guys only won 25%. But what did Ralph Steinman do to win 50% of the Nobel Prize? Well, he did some interesting work looking at a certain cell type in our bodies that we see here under the microscope. This is called a dendritic cell. So going back here, we see these dendritic cells. These are of the macrophage monocyte lineage. And here's a larger picture, a blow-up of this dendritic cell. And the dendritic cell has this capability to send an appendage into the lumen of the gastrointestinal tract. And this senses certain microbes and certain microbial components. And that changes the surface of the dendritic cell so that the dendritic cell interacts with undifferentiated lymphocytes. And this causes these undifferentiated lymphocytes to change into regulatory T cells or effector T cells. And it's very important to have a balance of the regulatory T cells and the effector T cells. If you have too many of the effector T cells, have a very high inflammatory response, which can cause injury. So, for example, injury to the gastrointestinal tract. If you have too many of the T regulatory cells, then you can have a problem with not being able to respond. So it's important to have this balance. And this is one of the things that these dendritic cells do. Okay, so that's mucosal immunology and how you have an interaction in the gastrointestinal tract with microbes. And we are beginning to recognize that most of the microbes in our gastrointestinal tract and other niches of our body are our friends. They're not pathological. These are actually components of our bodies that help us. So here, I'm going to just begin to shift and ask you some questions and ask you, amniotic fluid is sterile, true or false? Placenta is sterile, true or false? Meconium is sterile, true or false? Human milk is sterile, true or false? Well, prior to the microbiome period, the answer to most of these would probably have been sterile, that these different fluids are sterile, or these different tissues are sterile. But we're beginning to recognize that they are not really sterile, that there is a microbial component to all of these. Now let's start over here. This is a study that was done in 2008 at Stanford University. And in this study, they were able to get amniotic fluid samples from a large number of mothers that were stored in a biorepository. And using these amniotic fluid samples, they looked at the gestational age at delivery and the amount of microbial DNA. So x-axis is the amount of microbial DNA, and here's the gestational age at delivery. And they were able to find microbial DNA in the amniotic fluid. And there was a direct relationship to the gestational age. So more microbial DNA, the lower the gestational age. This suggests that the more microbes in amniotic fluid, the greater the potential for the development of prematurity. What they also found in this study is that those babies that had the highest level, or those babies who were born prematurely and had the highest amount of microbial DNA in their amniotic fluid, they also had the highest levels of pro-inflammatory mediators and white blood cells, so more inflammatory response. So that led to the hypothesis that perhaps that this inflammatory response and having microbes in the amniotic fluid might actually trigger premature labor. A few years after that, Dr. Kirsty Agard at Baylor University Medical Center did some studies looking at the placentas and found that the placentas actually contained microbes, microbial DNA. And there was also a relationship between the types of microbial DNA that was found and the level of prematurity, again, suggesting that there might be a relationship to the development of prematurity and microbial DNA. In our university, at the University of Florida, we did some studies, and these are two of my fellows at the time who were looking at the microbial DNA in meconium, and we found that that there was microbial DNA in the baby's first stools. And many of these microbes looked like the same kind that was found in previous studies in amniotic fluid. So this suggested to us that perhaps the microbes that we found in the meconium may have come from swallowed amniotic fluid. And one hypothesis that was raised is that microbes that ascend from the vagina can cross the choriodecidual plate, get into the amniotic fluid, and the fetus swallows substantial quantities of amniotic fluid, and then that is reflected in the meconium. There are other mechanisms as well. For example, translocation of bacteria through maternal gastrointestinal tract that may also lead to microbes getting into the fetus and also the meconium. This has not yet been clarified exactly where these microbes come from. But we're beginning to recognize more and more that the pregnant mammal, the pregnant human, is a symbiotic community. It's not a dyad with the fetus and the mother. It's actually a holobiont or a triad, which includes the microbes. These microbes, even if they're present in low quantities in the fetus or in the amniotic fluid, they are present in high quantities in the maternal gastrointestinal tract. And the maternal gastrointestinal tract produces large quantities of metabolites, so that about 35% of the metabolites in mammalian blood has a bacterial origin. And these metabolites can get to the fetus, and they have physiologic functions. And we're just beginning to clarify what some of these physiologic functions are. So, does the maternal nutritional microbial environment matter? And if so, how? Well, here is a study that was published in Science, in the journal Science, in 2016. And this was a study that was done in animals, germ-free mice. These germ-free mice were colonized with E. coli HA107 at day 10 of pregnancy. As you know, the gestation of mice is 21 days, and at day 10, these pregnant mice were colonized with one bacteria, okay? So they only had one bacteria in their entire body, and this bacteria only persists for 5 to 7 days. And then after the babies were born, they compared the inoculated to non-inoculated offspring intestinal innate immune systems. And what we see in these graphs on the right here is two different cell types that were compared. And at the bottom, we see the controls, and here we see the gestational colonized animals. And these are the pups born to these pregnant mothers who were colonized during pregnancy. And we see a greater innate immune response 5 days to 60 days after birth in both of these innate immune cell types. So this is telling us that microbes in the mother during gestation drive the innate postnatal immune development, a very important study. Now, what about the microbes found in the pregnant women during gestation? One question that was asked by a colleague, Amri Koren asked this question whether the microbes in the first trimester of pregnancy in the gastrointestinal tract of a pregnant woman differ from that in the third trimester. And it was found in this study, looking at stool samples from mothers from T1, which is the first trimester, compared to the third trimester. And this is something called a principal component plot. And here we see these different colors. Orange signifies the first trimester, and the gray signifies the third trimester. And you see in this principal component plot, each one of these dots here signifies a different microbiome from a different mother, that there's a separation. Here, the first trimester are obviously different than these here. These are samples taken from males and females, non-pregnant females and males. And you can see that these samples are also very different than during pregnancy. Then something very interesting was done with this study. They took these samples here from the first trimester and put them into germ-free animals, and nothing happened with these germ-free animals as they grew. But when they took these samples here, the third trimester samples, put them into germ-free animals, these germ-free animals ended up being fatter and had insulin desensitization, which is very similar to the phenotype in later pregnancy that you see in pregnant women. Many pregnant women develop insulin desensitization. And so this told us that these microbes have the capability to be transferred to mice and actually change the phenotype of the mouse. Very interesting finding. The next area that I'd like to just talk a little bit about when it comes to the microbiome relates to C-section. Here's a picture of the globe, and you can see different C-section rates in different areas of the world. You can see Egypt actually has a fairly high C-section rate. Brazil, Argentina have very high C-section rates. China, fairly high C-section rate. In certain parts of South America, the private hospitals in Rio de Janeiro or Sao Paulo, it's very unusual to see a vaginal delivery. There are various reasons for this, but we're beginning to recognize that vaginal versus C-section delivery may have some adverse consequences. It has been seen with epidemiologic studies that C-section delivery is associated with higher rates of autoimmune disease such as multiple sclerosis, type 1 diabetes, certain kinds of infections, and obesity. So how does this happen? Well, here is the two different modes of delivery, the C-section here versus vaginal delivery, and here's a hypothesis. With vaginal delivery, there's exposure to vaginal flora. You have normal microbial seeding of the gastrointestinal tract with these microbes that get into the newborn's gastrointestinal tract that induce normal development of immunity. With a C-section delivery, these babies are not exposed to the normal vaginal microflora, and so you have abnormal seeding of the GI tract and abnormal development of immunity. This concept was looked at about 10 years ago by Dr. Maria Dominguez-Bello, and a small study was done in Venezuela where they looked at the stool microbes of babies and skin microbes of babies born by C-section versus vaginal delivery. And here we see these red and these pink dots. This is, again, something called a principal component analysis. Those babies born by vaginal delivery, if you look at their stools, those microbes look a lot like the baby's mother's vaginal microbe. Those babies born by cesarean section, their stool microbes look not like mother's stool microbes, but more like the mother's skin. So that led to this concept that maybe we should be doing something to intervene. But before we get to the intervention, I want to show you another study. This is a study that was published in Nature more recently. And what this study showed is that it's not just the mode of delivery. If a mother delivers a baby by cesarean section versus vaginal delivery, there are confounding factors that also occur. Most babies who are born by C-section delivery also are exposed to antibiotics more because the mother gets antibiotics with the C-section delivery. Those babies stay in the hospital longer if they're born by C-section delivery. They also do not breastfeed quite as readily. And so here, what we see is the at the bottom, these different bar graphs. And again, see over time, the microbes differ in C-section versus vaginally delivered babies. But at the top here, we see that there are confounding factors. It's not just the baby going through the vaginal tract versus C-section that causes this difference. Okay. So one of the therapeutic measures that people thought might be useful here is to do some restoration of the microbiota of cesarean-born infants via vaginal microbial transfer. And the way this is done is that if a baby is born by cesarean section, you take a gauze swab and you put that into the mother's vaginal canal for about an hour. And you take that baby who is born by the C-section route and you take that gauze pad and you put that into the baby's mouth. You swab the baby's mouth with it to cause seeding of the vaginal microbes in the baby's mouth. And you wipe the baby's face and chest with this. And this is a paper that actually showed that you can restore the microbes in the gastrointestinal tract, the vaginal microbes in the gastrointestinal tract of these babies. Now, this was a very small study that showed that this is possible, but this is still not something that is recommended. And the American College of Obstetrics and Gynecology a few years ago recommended against this practice until better data are available. establishing the safety and benefits of this practice. So let's assume that the mother had herpes infection or group B strep, and you do this to the baby, you might actually be inoculating the baby with a pathogenic microorganism. So this is still something that is not yet considered to be standard practice, and it's actually recommended against doing this. Now, in preterm babies in 2012, the American Academy of Pediatrics looked at human milk and looked at the data from human milk and thought that it would be very important for providing baby's own mother's milk or donor milk to premature babies weighing less than 1.5 kilo. And they came out with this statement: if mother's own milk is unavailable in neonatal intensive care units despite significant lactation support, pasteurized donor milk should be used. And quality control of pasteurized donor milk is important and should be monitored. The problem with the pasteurization process is that you can see with this, and this is courtesy of Peter Hartman from Western Australia, that the pasteurization process kills a lot of the live components of human milk. And so you see lower enzymes. So for example, bile salt stimulated lipase is almost totally abolished. Alkaline phosphatase is almost totally abolished. IgM, IgA, IgG are markedly decreased. So many of these components that you see in baby's own mother's milk are demolished in the pasteurization process. So donor milk is not the same thing, not the same food as the baby's own mother's milk. We also have found, in terms of the microbiome, that there are human milk microbes. And prior to about 10 years ago, when we found human milk microbes when we found microbes in milk, we would throw that sample away. But we're beginning to recognize that these microbes are commensals and they are not necessarily pathogens. Here's one study that looked at the microbiome of breast microbes from 16 different women. Each one of these S's represent a different mother. And each one of these time periods here represent 1 week, 2 weeks, and 4 weeks after birth. And these colors represent different microbial taxa, okay? So in this one mother here, 1 week, 2 weeks, and 4 weeks after birth, the colors look similar. In this mother, 1 week, 2 weeks, and 4 weeks, the colors also look similar. What we are seeing with this is that the microbes differ markedly from one mother to the next, but don't differ that terribly much over time. So this is suggesting a personalization of the microbes in the mother's milk. So one mother's microbes are very different than another mother's microbes. Where do these microbes come from? Well, we're still not sure yet, but it's thought that most of the microbes come from the maternal gastrointestinal tract, either through increased translocation because the tight junctions are more permeable later on in pregnancy, or because these dendritic cells actually pick up microbes and take them through the bloodstream and carry them to the breast milk of the mother. We also are recognizing that the human milk and the process of breastfeeding and close contact between the baby and the mother is a very important process. That if the baby is exposed to the mother, you have this system called the enteromammary system. And the way this works is that with close contact of the baby and the mother, if the baby is colonized by a certain type of a microorganism, the baby's immune system may not be able to respond very well to that, but the mother's immune system does. So the colonization of the mother into her gastrointestinal tract, and she responds by making an immune response that gets into her milk, and that gets into the baby. And the baby actually gets protection through the breast milk, and this is specific protection. In the late 1970s, when this was first recognized in animals, but now we are beginning to get some studies in humans that show that this enteromammary dynamic system actually works. What is the number of microbes in human milk? This is a study from Spain showing that the number of microbes in the colostral phase, the transitional phase, and the mature milk are about 10 to the 5th to 10 to the 6th microbes per milliliter of milk. So that's actually quite a few microbes. And if we compare that to studies that have been done in terms of probiotics, if we assume an intake of 800 milliliters per day and 10 to the 6th bacterial cells per milliliter as found in these studies, this can provide about 10 to the 7th, 10 to the 8th bacterial cells that are personalized daily, which is close to the dose in most given to babies in most probiotic studies. So you are actually giving microbes that are personalized if the baby is being fed baby's own mother's milk. So where are we going in the future? Should we be focusing on the use of fresh mother's milk rather than bank donor milk? Obviously, I think that this would make a lot of sense to focus on that. And how can we promote mother's fresh milk use, and is there some way to improve on banked donor milk? Well, studies that were done by some of my colleagues at the University of Florida involved taking donor milk and baby's own mother's milk and taking small quantity baby's own mother's milk, 1%, 10%, 5%, and adding that to the donor milk, and then letting that incubate for 1 hour, 4 hours, 8 hours, and 16 hours. And we found that we could actually expand the microbes, live microbes, in donor milk by use of the baby's own mother's milk. So that suggests we might be able to utilize baby's own mother's milk to expand these microbes to get a microbiome more similar to that of the baby's own mother's milk. So this is an advance. I do not recommend doing this at this juncture, but this is something that with additional studies may be very helpful. We've also seen metabolites, and just recently published a paper on the metabolites showing that this technique can improve on the metabolites of donor milk. This is just a summary of our studies on transfaunation doing this technique. Each mother has a unique microbiota, and the live microbiome in donor milk can be restored with these unique bacteria using small amounts of mother's own milk. And this is a novel approach, and somewhere using a percentage of 10% and incubating for about 4 hours seems to be a reasonable restoration strategy. But again, a lot more work needs to be done for this. Now, I also want to talk a little bit about antibiotic use. And as you know, antibiotics are used a lot in mothers, pregnant women, and lactating mothers, as well as babies, especially preterm babies. And what do antibiotics do? Well, here's a cartoon that shows that without antibiotics, you have a balanced microbial community. This sort of looks like that coral reef that I showed you before. But if you give the antibiotics, you have a thinning out of the kinds of microbes. So the commensal, you may lose some of these commensals, and you get immune dysregulation. It's something called a dysbiotic microbiome. Do antenatal or postnatal antibiotics affect the neonatal microbiota? We are beginning to recognize that, yes, that is the case. Here are 4 different scenarios. Here is no antibiotics given to the mother or to a baby. Here is antibiotics given to just an infant. Here's different degrees of antibiotics. Okay, this is given to the mother, and this is to the mother and to the infant. Okay, so we see here that the types of microbes, these are phyla of microbes. This is the normal situation, but you actually increase the amount of certain kinds of microbes by giving antibiotics. The microbes that you increase are these phyla called the proteobacteria. Proteobacteria are microbes that consist of a lot of gram-negatives with lipopolysaccharide in their cell walls. And this can theoretically cause a dysbiosis to both the mother and the baby. So before I said that dysbiosis may actually lead to some problems. Here is the obesity antibiotic in the United States, and you can see that at the left here, we see the antibiotic prescriptions per 1,000 people, and you can see certain states. The darker the states, the area, the more antibiotic use. And on the right, you see the percentage of obesity. And there seems to be a correlation between antibiotic use and obesity. Again, that's a correlation. Now, what about disease that we see in preterm babies called necrotizing enterocolitis? Picture a baby with NEC. This is the radiograph, and here's a baby at the time of surgery. What about antibiotic use, and what do we see in the microbiome prior to the development of necrotizing enterocolitis? This is a study that I did with a colleague from the Baylor Medical Center, and we looked at the genome, the microbiome of babies from 8 different neonatal intensive care units in the United States, and looked at the different microbes. And these are control babies over time, and these are necrotizing enterocolitis babies over time. And we see this increase in the blue over time, but we don't, this is in the necrotizing enterocolitis babies on the right, but we don't see this increase in the control babies. This is telling us that prior to the development of necrotizing enterocolitis, there's an alteration in the microbiome. Could this be related to antibiotic use? Well, we have this association, and here we see the average length of treatment with antibiotics increases the odds ratio for the development of necrotizing enterocolitis. So if we give 5 to 8 weeks of 5 to 8 days of antibiotics after birth, that increases the odds of necrotizing enterocolitis by 50%. Again, this is an association, and it does not prove causality. And what we need to do in order to prove causality is to have better animal models. We need to have randomized trials, and we're beginning to recognize that this newly developed technique called multiomics may actually help us with this. Now, what is multiomics? Well, here is sort of an example of multiomics, but this comes from a parable from India. This is a Hindu parable that talks about the 7 blind men and this elephant. These 7 blind men were asked to go and feel different parts of this elephant. One of the blind men felt the side and said, "This is a wall." One felt the trunk and said, "This feels like a snake." The other one felt the tail and said, "This is a rope." And they were all adamant about what they were feeling. But they only felt a part of the elephant. They could not put the picture together. But if they would have collaborated and thought about this in a more collaborative way, they would have been able to recognize that this was an elephant. And this is where multiomics comes in. If you look at just the types of microbes in the gastrointestinal tract, for example, it'll tell you the names of the microbes, but it doesn't tell you necessarily what they are doing. But if you look at something called the transcriptome, the metabolome, you put these different omics together, you will then be able to tell what is actually going on. And there are now techniques where you can put these together and use systems biology approaches that actually show a more complete picture. It actually gives you more the complete elephant of what is going on. This is systems biology, and we are just beginning to develop the bioinformatics for these multi-omic approaches. And this is something that we will be able to apply to our premature babies, because one size does not fit all. The type of nutrition that we give to these babies may need to be very different for a 23-weeker versus a 29-weeker. And we are beginning to recognize that the kind of intervention, the nutritional intervention, or what we give in terms of antibiotics may actually affect these individuals in terms of adverse outcomes. And these adverse outcomes may not just be some of the things that we see in the neonatal intensive care unit, but actually beyond for the entire lifetime of this individual, and perhaps even transgenerational from one generation to the next. So I'd like to just mention that we have in the past nutritional pyramids, but we are starting to get away from these nutritional pyramids and beginning to think more about multi-omic approaches and being able to personalize nutrition for the individual using these multi-omic approaches. So I'd like to stop this lecture with some take-home messages. First, microbes play an important role in our overall physiology before and after birth. Perturbations in our microbial host interactions have lifelong and perhaps even beyond consequences. Well-done clinical studies along with newly developed multi-omic technologies will help us reach our goal of more personalized medicine. And I'd like to acknowledge so many people I have worked with in the past and who have helped me with my studies. And also, thank you very much, Shukran, for your kind attention. I'm happy to answer any questions.
Professor Joseph, thank you very much for your illuminating, impressive lecture. Actually, we felt during the lecture that there is another word of microbiome we are living with. We felt also the major importance of vaginal delivery in comparison to CS. And unfortunately, in Egypt, usually we have elective CS at 38 weeks, which is the usual. We rarely find babies even CS at 40 weeks. They usually do the elective section at 38 weeks, which causes a lot of harm, as we felt with your lecture. I don't know if you have the same issue in USA or it's unique for us in Egypt, this problem.
Well, I think that there are different places where there are various reasons for having more C-sections in different regions of the world. Okay, and in the U.S., my understanding is that we have about a 32 to 34% C-section rate. And we are trying to be a little bit more careful with elective C-sections. And I think that different parts of the world are beginning to recognize that this may have adverse consequences. So I think that, this is something that hopefully we will begin to recognize more and put this into our equation of potential harm that we can be doing to our babies.
Lecture to be attended by our OB and Gynae colleagues. I think it deserves to be given to them. Another point, Professor Joseph, for the enteromammary immune system, you mentioned that the GIT microbiome of the mother is very important for the colonization of the baby, which puts a big or an important question on what the mother eats. I think this is not this issue should be given great importance. I know, of course, that the microbiome is not only what she eats during pregnancy, it's what she eats since her infancy. But I think it's important to pay attention for the nutrition of the mother and the avoidance of fast food and many of the health tips that should be given and taught to mothers during pregnancy also.
Absolutely. I couldn't agree more. It's so interesting that some of the more recent studies that have looked at the metabolome of milk show that different parts of the world, so for example, in China, South Africa, Europe, you see differences in the in the metabolome of human milk, and you see differences in the microbiome of human milk, depending on region of the world. So these are different regions where the diet is very different. And so I think diet does play a major role in terms of the types of microbes that we see and the metabolites that are produced by the microbes, and also the metabolites produced by the mother. So yes, absolutely. I think that this is an emerging area that we're just beginning to learn more about. And junk foods and bad foods eaten by the mother will be reflected in the baby's diet. Absolutely.
Okay, I have a lot of questions. Yani kindly, we need also to talk about some points regarding the bovine colostrum. There are some papers emerging about the priming of the GIT of preterm babies with bovine colostrum whenever maternal milk is not available, and also donor milk is not available. And they are saying that donor milk usually is not colostrum because it's usually from a mother who is feeding for a while before her baby is a little bit older. And that bovine colostrum in a baby with no history of allergy to cow milk or family history, might be suitable for priming the GIT for the first 2 to 3 days until maternal milk is available. Of course, these are still researches, but I would like to know your point of view about it.
Yeah, so my point of view is that these are still very preliminary, and I think that it's a reasonable idea, and we're going to have to be very careful. From a theoretical perspective, I think the bovine colostrum is going to be nowhere near the baby's own mother's milk. And my preference is that if we are going to place a lot of resources into studies, I would like to place those resources into ways that we can nurture and help the mothers produce their own milk. So if they're born by C-section, this is a problem sometimes if they're born by C-section because the mother may not be able to make the colostrum for the baby as readily as if the baby is born by vaginal delivery. So, the things that we can do to provide the baby's own mother's colostrum, to me, make the most sense. But I think that the bovine colostrum studies are reasonable. They're still very early, and we'll see what happens with that story.
Okay. And for about the usually we have a baby, for example, recovering a premature baby and recovering from NEC. And whenever we want to refeed the baby, usually, of course, the first choice for us is the maternal milk. Sometimes we don't have the maternal milk, even after trying to enhance. And there is no also available donor milk. And in this situation, would you advise to give the premature formula as starting or refeeding a baby after NEC, or we can use the amino acid based formulas or the extremely hydrolyzed formulas that are available?
So that's a really good question. and there's some nuance to the answer, okay? And number one is it's very important to put food into the gastrointestinal tract. If you just provide TPN for prolonged periods of time, you will not have a trophic effect. You will not have growth of the epithelial cells. And I think to put some food into the gastrointestinal tract is very important, okay? Now, some of the babies who we think have necrotizing enterocolitis may actually have food protein-induced enterocolitis syndrome, FPIES. And those babies generally will have a bloody stool, okay? And what I do with babies that I think have FPIES, I use an elemental formula, okay? Highly hydrolyzed formula. But it's very difficult to tell if the babies actually had FPIES or not. The important thing is to put some food into the gastrointestinal tract, and I think to put formula in the gastrointestinal tract, just regular formula, is reasonable, okay? I would rather put food into the gastrointestinal tract than not put food in the gastrointestinal tract. If you use a hydrolyzed formula, look at the label and make sure that it is not something with a high osmolality, because the high osmolality formulas can actually cause damage to the gastrointestinal tract, okay? So I would try to stay away from the high osmolality formulas if you're going to give that as the feeding. But food in the gastrointestinal tract, I think, is very important. Okay. I see Tom Jacks is there. Hi, Tom. Yeah. Please chime in if you'd like to answer.
Welcome, Professor Tom. Professor Joe, another question, please. Giocal, which is starch-based formula together with lipids, and sometimes, or it is mainly used in inborn errors of metabolism when there is a problem in amino acids or protein metabolism. Giocal as a component can be added to the premature to premature baby to the maternal milk, as because of course in Egypt we don't have fortifiers. In this case, if the baby is not growing well and we are using the optimum or the full breast milk amount that we can use, and there is no anemia, there is no electrolyte disturbance, there is no sepsis, however, the growth is still not appropriate. Would we use Giocal, or you don't advise?
So I have to plead ignorance. I am not familiar with Giocal. I am familiar with the various fortifiers that increase the protein content and increase the calcium and phosphorus. And for the smallest babies, it's very difficult to get the amount of protein and calcium and phosphorus into those babies. And so if you have something that appears to be safe, and I don't know the composition of the Giocal is, and if you've had any studies to actually look at this. But again, I think that this is something that should be systematically studied. I know that with the fortifiers, we can improve the growth, we can improve calcium, phosphorus, and bone growth as well. So fortifiers are very important for our smallest preterm babies.
Okay, I have another question on the chat. What is the optimum time of MPO after NEC?
I don't know the exact answer to that question, but in general, what most people do, what they've been doing is going 5 to 10 days. But NEC is a very heterogeneous condition, and you have some babies with just slight distension of the abdomen who have some questionable pneumatosis and who have nice soft abdomens. I think some of those babies can be fed 2-3 days after the initial diagnosis. Obviously, if you have a baby who's had a very distended abdomen, who's been very sick, who's had a major inflammatory response, I would try to individualize that a little bit. We don't have a timing that is the magic number for feeding for babies with NEC.
Political answer.
Sorry.
Okay. Of course, we have lots of questions, but I know your schedule today is very busy and that we are taking part of your precious time. Thank you very much, Professor Joseph. Thank you for being with us. Thank you for sharing for the third time in series our meeting and conference. Hoping you a good health and hope to see you with us face-to-face next year, inshallah, in our meeting. Thank you very much.
I look forward to it. Thank you so much.
Thank you so much, Professor.
Dr. Safa?